glad you didn't forget to watch this last class on perceptual optics because we're going to be covering memory today. Here's today's course content. Plasticity seems to be how the brain stores memories, whether that is strengthening or growing new synapses, decreasing or removing synapses, and up and down regulation of receptors. And there's also talk about intracellular mechanisms, including regulating the expression of particular genes of DNA. Neurotransmitters are the molecules that carry the signal of one neuron to the next neuron. And generally it's done with these chemical molecules, but there are cases where electrical gap junction is used instead. But in general, it's going to be these neurotransmitters carrying the signal. This is showing a receptor or a ligand coming and attaching to a receptor on the next neuron on its dendrite, and it would basically open an ion channel, whether it was right there on site or slightly removed from the receptor itself. Once an ion channel has been opened, ions can flow through. So in this example on the right, we see potassium ions are coming in to that channel there that got opened. An antagonist tends to close up an ion channel when it binds to a receptor such that ions do not want to cross into the neuron, the next neuron. Agonists have the opposite effect. They open the ion channel and allow ions to go through, and so that tends to make the neuron more active or fire more frequently. These are some of the terms that are used to describe this process. So affinity would be the strength of binding between a molecule or ligand and a receptor. Efficacy would be the ability of a ligand to activate a receptor, or the intrinsic activity. Potency would be the degree of concentration required for a specific biological effect. And selectivity would be the degree to which a ligand binds to its target compared to other targets. Glutamate is the chief excitatory neurotransmitter of the cortex. GABA is the chief inhibitory neurotransmitter of the cortex. You also have several levels neurotransmitters coming from the brainstem, in particular like the ARAS, ascending reticular activating system. So you have serotonin, which is the longest lasting modulator, days to hours modulation. You have norepinephrine, which is going to be minutes to seconds modulation. Dopamine is minutes to milliseconds modulation. And acetylcholine is seconds to milliseconds modulation. Again, this is modulating level of wakefulness, but it also might have some effect on controlling or modulating frequency of context of consciousness. So what does memory have to do with perceptual optics, you ask? Actually quite a lot, because if we didn't have memory, everything would appear as an impressionist painting because nothing would have any meaning. So it's actually pretty critical. Memory allows the building of lifelong prediction models based on familiarity and past experiences, including movement and depth information. The right hemisphere dorsal stream would primarily handle the 2D aspect of color and location of pixels. And the left hemisphere ventral stream would primarily handle the 3D understanding of shapes, and with help from the prefrontal cortex would carry attention, logic, and meaning. And it would, in my model, include the bank as well as the imagination schemas, which can transiently model the real-world schema, but are usually conceptual. And the ventral stream can process shapes at any level of hierarchy, so you can pay attention to an entire scene, the shape of the entire scene, the contour, the topological profile of the skin or point cloud of the whole scene that you're looking at, or you could zoom into like just one particular item, like a writing pen or something, and just focus on the shape of that one thing. The hippocampus is so named because it is shaped something like a seahorse, and it sits at the top of the hierarchy of sensory processing, in particular your ventral stream. Its cortical structure is unique. It only has 3 to 4 layers as opposed to the 6 layers of the rest of the cortex. And it's organized as a bunch of series loops, so a bunch of series circuits in parallel. So you start in the entorhinal cortex, you go to the dentate gyrus, then to CA3, CA1, subiculum, and then finally back to the entorhinal cortex, and then back to the whole cortex. So you've got kind of this step-by-step process, instead of, the rest of the brain doesn't really work quite like that except for maybe the hierarchies. So it's kind of a very unique structure in how it's arranged and how information goes through it. It also is said that it receives timestamp clues from the entorhinal cortex, which in turn gets it from the medial prefrontal cortex, medial septum, subiculum, parahippocampal gyrus. But I think in general, the hippocampus wants to look for other unique aspects or features of each moment of consciousness. The hippocampus stores these unique aspects as abstract concepts to be reactivated upon encountering similar such things in the future. In the apparent absence of novelty for a particular moment of consciousness, it's still going to have its own unique timestamp. So it's going to be some time in the day, it's going to be some day of the week, some week of the month, some month of the year, some year within human history, some year within your own life, how old are you? So kind of this sort of timestamp that you get a gist of when you're saving episodic memories. There's also probably going to be some kind of spatial unique aspect, I would argue, in terms of the ring, and how things would be saved. Prefrontal cortex also plays a role in generating expectations and predictions based on past experiences and context. But yes, pretty fascinating region of the brain. It's kind of boggled people why the hippocampus deals with both space and memory. In my model, though, I think it would be kind of clear why that would be the case, because perceptual space is fundamental to experience, and memories are trying to record the gist of experience, so it needs to understand space too. If you've never heard of this term, long-term potentiation just means the strengthening of brain connections through repeated stimulation, leading to improved memory and learning, and the hippocampus definitely is implicated in the process of memory and organization. Okay, this is a very confusing diagram. Let me try to explain it for you. You have what are called first-degree neurons and second-degree neurons and third and fourth and so on. So, let's say that in your eyeball, the retinal ganglion cell neuron is the first degree because it's carrying information from the eyeball to the lateral geniculate nucleus. And then there will be a second-degree neuron that carries that information from LGN to visual cortex, primary visual cortex. And then there will be a third-degree neuron from there carrying it to a higher order visual region. So you've got these steps. So if we consider that there was maybe 20 neurons all converging onto 20 first-degree neurons all converging onto a second-degree neuron, and we want to consider the timing of when the action potential of all those previous 20 is coming to the second-degree neuron. This dotted line here in the middle is the timing of the second-degree neuron action potential. The dots are the arrival times of these various first-degree neurons. So if they get there just a little bit before the action potential fires, then the second-degree neuron wants to reward that behavior by upregulating the AMPA receptors in the connections with those neurons. And if it's late to the party, it wants to punish that behavior by downregulating. And what's confusing here too is that the past is represented out to the right, and the future is represented out to the left. Kind of counterintuitive. This slide needs just a little bit of clarification, which is to say that we really don't know exactly how memory works, but an idea with sleep is that particular sequences of ensembles of neurons are repeated at a faster rate, for example, 10 to 20 times as fast. And they've actually recorded this, for example, from rodents that are going in a maze, learning a new maze. They see certain ensembles of neurons in a particular sequence and at a certain rate. And when they fall asleep, they notice that the same sequence is repeated, but at a much accelerated rate, like 10 times as fast, 20 times as fast. Now, what that could be doing is bringing a 100 millisecond cycle of consciousness, shrinking it down to 10 milliseconds. Of course, it would still be post the next action potential. So looking back at the last slide, it wouldn't really make any sense. It would seem like it would be down-regulating. But you also have neurons that fire a little bit kind of early bird, like before the rest. So it may be that by compressing the time of, cycles of consciousness, that you can actually get a little bit of long-term potentiation from some of those early birds that is able to, either offset or just make enough connections on their own to make long-term potentiation. Still an area that they really don't know, but just an idea. These are your levels, neurotransmitters, and when you're awake, they're all online. So whether that's acetylcholine, serotonin, norepinephrine, histamine, orexin, they're all going. The brainstem, they're shooting up these neurotransmitters into the cortex. When you fall asleep in slow wave sleep, they slow down quite a bit. And when you enter REM sleep, most of them go completely offline, but you get this one, the acetylcholine nuclei, and in other words, pedunculopontine nucleus and lateral dorsal tegmental nucleus in the brainstem, are still sending neurotransmitters to the lateral geniculate nucleus, and from there onto the visual cortex. And so this seems to be, maybe modulating or driving the content or the context or the frequency of the context of dreams. So pretty fascinating. This is a very good diagram showing you your levels neurotransmitters. As you can see, they pretty much start in the brainstem, whether we're talking acetylcholine, dopamine, norepinephrine, serotonin. Although I will note that for acetylcholine, you have the septal nuclei up here, and you have the nucleus basalis right there in the basal forebrain. But in general, it's going to be in the brainstem, and it's shooting. They're shooting their axons to the cortex and releasing these levels neurotransmitters in the cortex. You also see here the substantia nigra involved in dopamine. This is the region that goes bad in Parkinson's. They get less dopamine, and then their movements are harder to initiate and harder to stop, and so it leads to the kind of back and forth oscillation of their arms and things like that. Unfortunately, what else do we have? We got Raphe nuclei for serotonin. We have locus coeruleus for norepinephrine, which is the same as like noradrenaline. Yeah, pretty fascinating little diagram. These authors here suggest that instead of considering like prediction and memory, we should look at 0.5 seconds into the future all the way to 0.5 seconds in the past as being a sort of time window. And in my model, I explicitly call this a flowing time window. In other words, you've got this future to past gradient represented by an axis within perceptual space in the fusing schema. Like the imagination schema, this fusing schema and flowing time window is mostly subliminal but I still think it drives your kind of sequence understanding and even the feeling of a lot of qualia, such as coldness or sense of self or mood or happiness. A lot of these things, I think, are vibrations on the ring that are carried with this flowing time window. And in this window, the future is always moving into the present, replacing it, and present is moving into the past and replacing it. And so you're continually get this future going towards the past, and that makes you feel like you are constantly moving into the future. So any of the qualia that have this kind of flowing sense to them, I think, are going to be largely found in the flowing time window. These are some good quotes. One of them says, "It is certainly reasonable to describe perception as extending into the past and the future, but note that to do so violates the accepted doctrine that perception is confined to the present." So it's this idea of a buffer or duration of working memory, like we were talking about in another class with Badley. In the flowing time window model, sequence is attributed to experience, not just to memory. So you're all the time, in any given millisecond, or any given 100 milliseconds, like one cycle, you are looking at a whole second of experience. You're looking into the future a little, you're looking into the past a little bit, so you're all the time seeing an entire sequence. Even though it's just a short duration, you're still seeing a little bit of breadth. It also says here, if perception involves the apprehension of a changing world, not a frozen one, the problem is that of detecting invariance under transformation. He also says the present is no knife edge, but a saddleback with a certain breadth of its own on which we sit perched, and from which we look in two directions into time simultaneously. The unit of composition of our perception of time is a duration with a bow and stern, as it were a rearward and forward looking end. In my model, not only is there this flowing time window, but it can assume three different forms. So in the last slide, we were considering it in the terms of kickout. In other words, the ring would be, present tense ring would be stationary, and future would be flowing in and replacing it continually, and present would be flowing to past and replacing the old past, and it would be continually being updated as time in the physical universe progressed. But there's also a couple other things. So if you are focused on some object on your right and then you move your attention to an object on the left, well, while your attention was shifting, like let's say while it was in the middle between the two objects, your past was associated with the object on the right, and your future is associated with the object on the left. And you're kind of considering all three positions at once while you're at that one point of time. And so in that sense, future and past would take up spatial locations in the real world schema. Another, that would be like walk. And then another form would be self-motion, which is depicted here. So if a person is walking down a trail, their body schema is moving relative to the real world, or let's say to the environment schema to be specific. And so the future would be associated with a more forward spot on the trail, and the past would be associated with a further back spot on the trail. And so you can basically use the movement of the body to be looking from past to future. And I think that the brain actually does that in the creation of perception. If you had coronal rings going around your body, perhaps in the body schema or something, and you're walking down a trail, then the path that would leave would actually be a helix path. So a ring would become a helix when you have some kind of axial offset, whether that is by self-motion down a trail, whether it's by walk of attention, whether it is by artificial kick-out, like when you're listening to music or something. So when I speak of like a ring, I could be also kind of speaking at a helix, or speaking of a helix, depending on the frame of reference. So I'm not exactly sure how I would describe the flowing time window, but I do think there is a gradient of opacity. So you start at the present moment, and you're seeing into the future a little, you're seeing into the past a little bit. And you can even notice how the arm is changing from frame to frame to frame. And if you imagine that there's just this continuum of frames, you can start to see how you could actually use the body schema, the shape of the arm and the hand as it's changing frame by frame to actually guide the or be tied to the muscle commands in the motor cortex that actually make it happen. And just as David Eagleman speaks of, like, speech babbling when you're a baby to learn how to speak, and you're just making sounds and seeing what certain neurons do, or certain, you take some action, you see what happens. What did it sound like? And you're trying to imitate what your parents sound like. And so once you get close, you're starting to learn better. The same thing could be like motion babbling. Like, you start moving your arms around and see how it works. So you can use kind of the real-time sensory input from your eyes that's coming back and telling you, hey, what did this do? This action that I just sent out, what did it actually do? So the idea of a flowing time window is basically the same thing as working memory. It's the brain's perception or conscious perception of time passage. All right, for what it's worth, I have 100% confidence about all the other underliers of consciousness that I've mentioned in this class, whether it's the bank, whether it's rings, whether it's planes, frames of consciousness, all that sort of thing. I have 99 to 100% confidence in. The flowing time window, I have confidence, but I'm just not sure if I've got my head around it exactly correctly. And of course, the correlates for these things I don't have a lot of confidence on. Maybe 10 to 40% confidence on the neural correlates or mechanisms underlying these things. But the flowing time window, I do want to propose here that it's possible that this alignment tracer is not just an aligner, it's actually the print head of all of consciousness. Now, the idea is that science itself says that, hey, action potentials are painting consciousness. They are the thing that underlies consciousness. And so I'm really kind of just saying the same thing, just with a little bit more exploration of the exact mechanism. So the idea is that, like an old TV cathode ray tube would shoot the electrons in rows from left to right, left to right, left to right, and it'd be jumping down a row every time until it illuminated every pixel on the TV screen. So I think it's possible also that the colors of consciousness might be refreshed in a similar fashion, but with rings generally. So it would be like the top ring, then the one underneath it, and the one underneath that, the one under that, until it gets down through like 1,000 rings and gets to the bottom. But because it's going so quickly, maybe it goes through that whole cycle 100 times a second or something, or 50 times a second. Therefore, it feels to you as though it's continuous. So, obviously speculative, but just throwing that out there that it could be, I would entertain exploring that a little bit. So it is inevitable that there must be delay. We have sensory input that takes 100 milliseconds to get to the posterior parietal cortex, whether that's vision, sound. Sound's a little bit faster, but proprioception information. It takes a little while to get to the region that is thought to be, providing consciousness. There's also roughly a 100 or 150 millisecond lag from that point until the signals from the motor cortex go down to the muscles and cause them to engage and make you start running or something like that. So you've got this lag like pretty much all the time. So I think what the brain might do is map the motor output to a region in the flowing time window that is at 0.2 or 0.25 seconds in the conscious future. In other words, the conscious present lags actual present by 100 milliseconds or 150 milliseconds. But starting from that what's called conscious present, you would go in 0.2 or 0.25 seconds into the conscious future, map that location on the time window to your motor output so that you always get the the right timing, and you don't get this lag between what you think and then what your body actually does. This is just showing you the trisynaptic hippocampal loop. So you start at the cortex, you bring information to the entorhinal cortex, and then you start the loop, go to dentate gyrus, then CA3, CA1, subiculum, back to entorhinal cortex, and then you can go back to the rest of the cortex. So the hippocampus is all the time going through this loop, running this loop. And then we're going to look in the next few slides about what these various regions may be doing. So this is the scientific explanation in terms for it. Dentate gyrus is doing what they call pattern separation. It's the first step in the hippocampus. It's looking for any and all features of current experience, whether that's unique novel features or whether that is pre-experienced features that you're already familiar with. CA3 would be involved in auto-association. In other words, if some abstract thing has been noticed or experienced before, allow it to trigger memories that share that commonality. And the list of memories that you would pull up would be what I call a skewer. Finally, CA1, pattern completion, would select the most salient or useful or applicable memory from that list. Generally, it's going to be the most recent item on the list. And go ahead and replay 1 or 0.1 seconds of that memory, like just as a little snippet video that you throw into consciousness. So you switch from paying attention to the real-world schema for 100 milliseconds and you're paying attention mostly or entirely to this little video snippet. Of course, the time range could vary a little bit. This other stuff would happen just incredibly quickly, pretty much too quickly for you to ever notice, unless you're just really paying extremely close attention. You might just barely see some of that stuff. So this is not just a analogy here. I am proposing that the brain actually uses perceptual space to lay out a stack of memories. So up at the top would be the most recent memory of seeing the letter B in this example, and then it would be going down until at the center is like your childhood when you're first learning how to write the letter B and memories associated with that. And just like, well, this is part of the imagination schema. So, like other components of the imagination schema, it's largely subliminal. It's very low opacity. You usually don't notice it unless you're just really trying to pay attention. So I think it's not outlandish that it would use perceptual space to help lay out the temporal sequence of a set of memories that's skewered by some particular commonality. Something like the letter B has an actual shape to it, and so it could have a shape to the skewer as it's going down and pinning a set of memories together. Other things would be perhaps too abstract to have any particular shape, but you could still have just a single line going through and pinning a set of memories together by some artificial commonality. This is just reiterating that the skewers that I propose would be viewed by the mind's eye in the imagination schema. As far as I know, this concept here is unique to me, but I think it's a concept that should be considered. And so basically it's the idea that the hippocampus takes about 1 month for any new feature that's been observed in experience to teach the cortex to be able to trigger that commonality or that feature or that episodic memory on its own without any help. And the reason I say that is because there was patient HM, Henry Molaison, who had his hippocampus removed, and he lost memory of any episodic experience up to 1 month prior to his surgery. So anything that happened 1 month before his hippocampus was removed, he forgot, which tends to suggest that it takes the hippocampus about a month or two to kind of replay memories and reiterate them before the cortex can handle re-triggering them on its own. And what the slide here says is episodic memory is primarily stored in the cortex. That's another, that's fairly, a large number of people subscribe to that. But for memories to be re-triggered upon encountering a new instance of some commonality, the commonality must be recognizable. The hippocampus repeats and potentially accelerates memory replays and links the commonality to existing memories, embedding the ability for the cortex to trigger appropriate memories in the future. So my idea is that the hippocampus isn't really storing the memories themselves. It's looking for features, novel features and familiar features, categorizing them, helping to store them, linking them to episodic memory, and kind of re-triggering and replaying those things that are mostly stored in the cortex until the cortex itself is able to trigger those things. And so the whole concept is the importance of what I call triggers, or features, basically. Anything in experience that is some feature, some unique feature, whether novel or whether familiar. In terms of episodic memory, what are the duration ranges and what are the possible correlates? Well, you have the working memory, which is the shortest, 0.1 to 10 seconds, analogous to what I call flowing time window. And its correlate would probably be the prefrontal cortex and the posterior parietal cortex. When you get to medium-term memory, 0 to 30 days, or basically 10 seconds to 30 days, you're going to need the hippocampus because it's doing this pattern extraction, categorization, storage, recall, replay, acceleration, all this stuff that's crucial to being able to re-trigger a fledgling memory trace in the cortex. For long-term memory, 0 to 10 years, the hippocampus has taught the cortex how to re-trigger that stuff. And possibly there's also this long-term depression in cerebellum. The idea there is that the nucleus, or the nuclei of the deep cerebellar nuclei, are sending output to the cortex, and those neurons want to fire, a little bit, but the Purkinje cells of the cerebellum are all the time inhibiting them. And so what you do is you long-term depress certain of those Purkinje cells, then you're allowing basically certain of the neurons within the deep cerebellar nuclei to fire more often to the cortex. So it's kind of like two negatives gives you a positive. So long-term depression of Purkinje cells in cerebellum may actually be somewhat equivalent to long-term potentiation in cortex, just a little bit different variety of it. If you're driving home from work, there's probably a path that you usually take, and the basal ganglia tends to learn sequences of motor and even just seeing landmarks, like, okay, here's this gas station, I'm going to turn left here. And so even if you've got in your mind that you want to stop at some store on the way, sometimes you'll find yourself driving home and forgetting about it, because the basal ganglia is good for like just autopilot muscle memory. And whether that's driving, whether that's playing an instrument, any kind of muscle memory seems to be largely implicated in the basal ganglia. But as far as like if you have some kind of mental map of your city that you're familiar with, and you do succeed in going to the store and not forgetting, then you probably used your hippocampus in that, basically that map to guide your driving. You stayed focused on your task better. David Eagleman has a very interesting concept. He calls it the neural pace layer memory system, something akin to a pace layer system of another person. I forgot his reference there. But the idea is that you have a fast changing layer at the top that updates anytime it encounters a new pattern. You have a moderate changing layer underneath that that updates if it encounters a particular pattern in the fast changing layer repeatedly. And then you have a slow changing layer at the bottom that only updates if it keeps encountering a particular pattern in the moderate changing layer. So one person was describing it as like, sediment shifting around and stuff. You've got like basically this range of paces at which you're storing memory. Certain things have to be activated multiple times to make a difference here, or a few times here, or just one or two times make a difference here. So that's a pretty interesting concept. How it actually does it, this is probably a pretty poor depiction of rough depiction of what he's arguing, but he did mention this stuff more or less, at least the stuff up here. So, and he did say that it may be that long-term potentiation is not the whole solution of storing memories. There may be these other things like gene expression, of course, up and down regulation of AMP receptors, whatever, that play a role in memory storage as well. As far as I'm concerned, I would consider probably 4 layers of this pace layer memory system. At the very top, the fastest changing layer would be the prefrontal cortex and posterior parietal cortex working memory, the flowing time window, because it's just kind of like the current second or so. So that's the very tiniest time frame you could work with. Then underneath that would be still a pretty fast changing layer, the hippocampus dentate gyrus, looking for novel and familiar patterns to link to episodic memory. Still in the hippocampus, you would have the moderate changing layer, and this would be CA1, recalling or replaying particular memories if they are salient. So you're starting to see this thing where, okay, if I'm encountering it a few times, or if it's salient, then I'll go ahead and replay it. You're starting to see this kind of moderate pace. And then the very slowest would be the cortex. Once the hippocampus takes 1 month of training in it, and it pulls out all the particular things that you learned in that month of life, whatever it thought most salient, whatever it encountered the most, or thought is most important, it's going to teach the cortex through dreams and through replay and acceleration and linking of an abstract concept to old memories that share that abstract concept somewhat. So that's how I would kind of look at this neural pace layer memory system. All right, I haven't talked about this yet in this class, but it's a concept that I do think is important, and it's this idea that the bank can kind of piggyback off itself. So if you get like 2 copies of the bank and they can overlay each other in the same 3D space, and let there be like a sphere selected off of one that is considered to be solid, a rotor if you will, and let another sphere cavity be hollowed out, a larger sphere cavity be hollowed out of the other copy of the bank, and let these two, let the one roll around within the other just like a spirograph, but in 3D, because it's one sphere inside a sphere pocket. And basically there's one point that it's always touching. The rotor is only touching the stator cavity at one point. And so that would be like the zero-dimensional tracer, alignment tracer. And as the spirographic motion happens, there's a ring path that is traced along the basically the transient intersection of the 2 copies of the bank, the rotor against the stator. And this would allow you to kind of 3D rotate the orientation of the rings, and also you could change the size in real time, because either of the bank could grow or shrink in real time. It would be a smooth kind of process, more or less. So there's kind of like an inertia to the change, if you will. But these are just kind of some generations by AI of art that might kind of depict it. I don't really feel like it did a great job because I'm sure you remember what the bank structure looks like, and it didn't really depict that in the first place. And it was supposed to be clear and colorless, so most of these are not clear and colorless. And so not very good depictions, but they are kind of pretty, so I left them in there. Um, but yeah, so it's depictions of the concept that the brain might piggyback its own structure off of itself by changing connections, pretending to be turning a rotor copy of the bank inside a stator copy. Uh, so it's just kind of like, how does the brain make perceptual space? It's a very tricky question, and maybe this is kind of how it pretends that uh, it's actually creating perceptual space. And really all it needs is a mechanism that will be faithful to a shared perceptual space so that it can carry out its calculations with that. So kind of interesting concept there. Let's go on to the next slide. So the brown sphere is the rotor, and the white sphere is everything inside the white sphere is hollowed out. And if you imagine there's more solid bank structure going outwards from that white sphere, then you've got basically the one instance of the bank rotating within the other. Assume there's no gravity here, so it can just go up onto the ceiling just fine and come back around. And so you get this kind of spirographic rotation of the one sphere inside the other, tracing these ring paths. And the green would be a depiction of the ring path. So the concept is that the brain may continually be making and breaking links between ensembles of neurons representing different voxels on a single bank structure, but it would be like two copies. And it would be sort of drawing the 3D rotation of one relative to the other in order to begin to create an understanding or a mechanism for handling a shared perceptual space. Over time, a spirographic movement is noticed, but the point at which the two copies touch, the inner solid sphere touching the larger hollowed cavity sphere, is the alignment tracer, and the path followed is a ring path. Either bank copy can add or shrink size, either one of them, so as to create a squiggly ring path, or they could stay the same and draw a smooth ring path, or to largely grow the ring path in real time to bring attention to a transiently distant stimulus, such as the sound of a car driving in the distance. So as this one sphere is kind of rotating inside this other, as you hear a car, it can grow in size very rapidly so that the touching point or the alignment tracer goes all the way over to where the car is to draw the understanding of where the car is at. But as you recall, the bank also has a cylinder inside, not just the sphere, and that can also exhibit spirographic activity, but it would now be constrained. The rotation or spirographic movement would be constrained to one particular rotation axis. And I do think that the brain does this sometimes. So here you've just got depicted like a coronal instance on the left. You have a sagittal, I think, instance here in the middle, and you have a transverse or horizontal example on the right. So basically it's further proposed that bank against bank can be made to follow cylindrical spirographic motion by use of cylinder within bank. Wheels, like the wheels on a car, follow such a type of rotation, but it may be that sometimes the brain simply samples from perceptual space in such a sweeping motion that holds a fairly steady axis. So it might be like kind of, if you're looking ahead of you, cycling from the scene in this sort of circular manner and spirographic, like one copy of the bank doing a kind of spirograph. I can't show it with the little cursor here, but I think you can envision what I'm talking about. Of course, it can switch in real time from cylinder to sphere and back, and grow in size, shrink in size, any of that stuff. So the use of the bank is proposed to be malleable in many ways. So here you have a first degree neuron and you have a second degree neuron. The second degree is called postsynaptic. The first degree is called presynaptic. And a lot of times we'll just throw away the degree completely and just talk about presynaptic versus postsynaptic. At the end of this first neuron axon, you have these little branches and you form synapses with the dendrites of another neuron, the postsynaptic. Right here would be the action hillock. So once you exceed a threshold at this location, there's no going back, and there's going to be an action potential carried all the way down that axon to the next neuron. Most receptors are prograde receptors, meaning they are located on the dendrites of a postsynaptic cell, but there's a few that are actually retrograde receptors, CB1 being one of those, and they are on the presynaptic neuron at the end. And basically when you, like if you get anandamide or something plugging into CB1, you're going to release less glutamate out of here. And when you release less glutamate, there's less plugging into the dendrites receptors on the next neuron, and so it's kind of an inhibitory effect. This is a chart I made. It is showing you endogenous ligands or neurotransmitters and the receptors that they tend to plug into compared against exogenous molecules, things that like coffee. If you were to ingest, the caffeine is going to antagonize the receptors for adenosine. Or if you drink alcohol, you raise the amount of GABA, you lower the amount of glutamate. If you were to drink St. John's wort tea, it's a reuptake inhibitor, which is kind of the same thing as increasing the molecules of glutamate or the molecules of serotonin and dopamine. Coffee plugs into the adenosine receptors and antagonizes them. In other words, it closes the channel, and so the neurons are going to be less likely to fire. But adenosine is a kind of like sleepy circuit, so when you antagonize the sleepy, you're actually making yourself more awake. You also have things like barbiturate, phenibut, fly agaric, mushroom. Can be, it's fairly toxic. It has some particular toxins in it that can be removed, but in general, as it is, the mushroom's kind of toxic. But anyways, it agonizes GABA receptors, but GABA is an inhibitory circuit. The channels, the net effect is that the neuron will be less likely to fire or fire less frequently. So agonizing GABA is kind of the same as antagonizing glutamate. So in both cases, you are taking 50 hertz brain activity that is more local, and you're slowing it down. You're throwing a kink, and so it slows down to like 12 hertz or 2 hertz. And because it slows down, it's able to resonate with a larger area of the brain, and you get more global brain activity integrated together, but you have like kind of these gaps and stuff. So a pretty interesting chart there. Okay, this chart is showing you endogenous neurotransmitters on the left, and then false neurotransmitters or exogenous on the right. Of course, some of these things can be dangerous. Also, some of them are penalized or whatever, what have you. But basically, it's just showing you the structure of what these are, and it's showing you also lipophilic or lipophobic and lipophilic ion channels, GPCRs, things like that. So kind of interesting. There is this concept called downregulation. For example, if someone smokes for the first time, they're going to get a lot of increased stimulation at the nicotinic acetylcholine receptors, but the brain will probably downregulate the number of receptors on dendrites if you keep doing that, because it's basically just a mechanism to avoid um, like overstimulation of neurons or something to that effect. Some of the serotonin agonists work by upregulation and also by possibly being capable of passing through the cell membrane to get to the inside of the neuron. There's also this concept of changing like brain networks and stuff via NMDA antagonists, so whether that's ketamine or what have you. Also, any kind of CB1 agonist would cause, I'm not sure if I'd say downregulation in AMPA, but I guess in old rut circuits at least, so that you're no longer rushing to do the same old thing that you always did. You actually may step back and kind of reconsider if that's really what you want to do, or something to that effect. So obviously it could be useful for like PTSD or something like that. The serotonin agonists seem to be more with like the making of new connections versus so much the breaking of old routines or ruts or PTSD or false fears that have been learned but are no longer necessary, things to that effect. Thanks for watching this course. Last slide is just kind of recapping again on this concept of NMDA antagonism, which is thought to underlie cyclization, both of neural action potentials and also of subjective experience. And also at particular levels of antagonism, you may get dissociation. Again, this reference here is pretty fascinating. Like I said, I have subjectively observed enough to be 99 to 100% confident in pretty much all of the underliers of consciousness that I've described in this course. The only one that I'm not exactly sure I'm describing correctly would be the flowing time window. It's a pretty difficult concept to get your head around, and I'm not sure I'm totally understanding it. As far as like correlates of these things, obviously, like I said, I'm less sure of the mechanism and correlate, but I am pretty doggone sure about the underliers of consciousness as I've discussed them in this course. So hopefully you found this useful, and thanks for watching.